Thanks very much, Deborah. And thanks for the invitation. So when I was asked to fill in for Ken, I was wondering what I should be able to tell you or how to organize this. So what I like to do is actually build up the case to the portfolio and really share the portfolio towards the end of my talk. And so I'll start with this slide that I don't think I need to convince anybody in this room that we are in desperate need of faster or shorter, safer, and more universal regimen. So we need more new drugs. And to top that, the output that we've had thus far has been quite bleak, as everybody in this room also knows. The last two compounds that have been registered, bedaquiline and delaminid, only came some 50 years after the last one, rifampicin, that was approved for the treatment of TB. And the last regimen that was also really approved was in the early 80s when rifampicin was really shown and proven to have great sterilizing activity with treatment shortening. So what I'd like to do a little bit is talk to you a little bit about the challenges that we faced, why this dearth of activity, and then look forward based on the last 10 years of activity in research, in basic research, and try to convince you why we believe that we're well poised over the next 5 to 10 years to really deliver some novel compounds for clinical trials. So why have we not succeeded thus far? Well, the most important is this lack of investment in TB biology and drug discovery over the last 50 years. I mean, that's just the bottom line. But more so, it's really, well, in parallel to that is our lack of understanding of the biology. So how the bug works, how TB works, under what conditions, in what environment. There's a lot of unanswered questions that really started only recently to become relevant to us. We've had very few validated targets over the years. The assay conditions were poor, and you will see a little bit later why we believe so and how we've remedied that. And the safety of drugs potentially have limited also the uptake and could potentially limit the use of really good drugs that we come up with. And then over the years, of course, we've had resistance come up against two of the best, potentially the best sterilizing and treatment shortening agents, rifamycin and pyrazinamide. And this has also been a big challenge for us. So what do we need to focus on? Given everything that I've told you about the challenges and what we've faced, there certainly need to be worked on novel mechanisms of actions, targets that have not been exposed to drugs thus far and therefore not subject to resistance. So that's the key, that's the first key, really novel targets, novel mechanisms. And I will go even beyond that, not just novel mechanism, but novel pathways. Not every pathway in the TB, if you will, world has been thus far examined. There are pathways that have not been at all looked at, and no compounds have been generated against those pathways yet. So there's a lot of unknown. Where is the best place to tackle the bacterium? In the granuloma, in cavities, inside macrophages, under what conditions? In aerobic conditions, anaerobic conditions, there's a lot of different ways that one can look at the bacterium. And probably all of the above will make a difference as far as what we come up with, the best regimen at the end of the day that we will come up with. And given where they are, and we know when we treat bacteria with a given drug or regimen, we can have a very fast kill, but the bacterium somehow adapts and goes into a more tolerant state. And what is that tolerant state? This is the kind of things that we need to tackle, understand better, and tackle as well in a really good portfolio. And finally, we do have well-validated clinical targets. We shouldn't forget those. Like I said, some of them have been hampered by resistance. If one could come up with a rifamycin analog that doesn't hit the resistance that we've seen in RNA polymerase, this could be an amazing find, for example, because that is a truly sterilizing compound. It's a truly treatment shortening compound. If we could come also with safer drugs to administer so we can increase the dose and potentially shorten treatment that way. And this is really the kind of strategy that pharma companies have used for a long time is to come up with follow-on compounds that are potentially better and safer and more effective than the first analog that has been developed. So we believe that really a combination of all of these will be necessary to create a really well-balanced portfolio that could develop and deliver over the next 10 years or 5 to 10 years. And hopefully I will convince you at the end that we're well poised to do that. So over the last 10 years or so, what's happened? The first is a downer, obviously. Some pharma companies have exited the field of TB, but that's not all lost, because the assets that actually were being worked by, let's say, AstraZeneca and Novartis have been transferred to the TB Alliance, so we are continuing to work on them and develop them and take them through as far as we possibly can into preclinical development and hopefully into development. So that's not completely a negative. But the rest are actually incredibly positive. There's been a huge and significant investment in resources, and primarily really by the funding and the investments that the Gates Foundation has made, and I'm going to go over a lot of that in the next few slides, and I think that's been incredibly helpful. There's been significant enhancement in academic research and discoveries over the last 10 years that really are helping us with new targets, new validated targets, new mechanisms of how to validate targets in genetic validation, for example, and those are very important, and also new chemotypes that are starting to emerge as well in the literature. We've also enhanced and prioritized the PDP portfolio, our own portfolio, and I think that's also very well poised to deliver. And of course, finally and lastly, the creation of the TB drug accelerator that everybody's talked about in the last session, or some people brought it up at the last session. And this is what I'd like to spend a little bit more time on, because I think through the investment that the Gates Foundation has made in creating the TB drug accelerator, whereas at the beginning it was really designed to bring some companies, pharma companies together to get access to a much greater pool of compounds and chemotypes, if you will, it really has evolved into a consortium of amazing capabilities, tools, and also expertise and knowledge from the various pharma companies and academic institutions and others that really are coming together to create a great portfolio of projects that could deliver. So this is the 2012 is when it was generated, and it brings, as I said, a number of pharma companies, PDP, which is us, and research institutes, and it really creates, as I said, a consortium of amazing technologies, capabilities, and tools and platforms, all under one umbrella. And this is a bit of an outdated slide. There are new members that continue to join to bring in new capabilities. But let me start from the left-hand side, upper-hand side, and I apologize for the people on that side, a little bit too far. I can't point, so, I'm only looking on this side because it's easier for me. It's closer, sorry. You have a huge amount of disease expertise that have now been brought together, from NIH to academic institutions, et cetera. And this really has heightened our awareness and our understanding of our lack of understanding of the bug. So I think it's really is helping us to ask the right questions and potentially come up with really excellent tools that we can then study the bacteria. On the animal model front, if you move to the right, we continue to expand our capabilities in the animal models field, especially in the mouse model, where we've also added Anne Leonard from CSU for the mouse model. Anne and Eric have also been working together under the resourcing of the foundation as well, or help the foundation to develop an alternative mouse model, which is a crumb-neck mouse model that develops cavities, which may mimic a bit more the pathology of the human disease. But at the same time, Cliff Barry is developing the marmoset model at the NIAID, and this is also undergoing further validation. So we continue to try to validate these models as best as we can so that we can have a better idea of how these models can predict treatment shortening ultimately in the clinical setting. Screening capabilities, which really are the key component here. As you know, we need to screen millions of compounds because of all the pharma companies. But between Caliber, which just joined, they just joined the TBDA, This is the organization that was created by Peter Schultz in California, with NIH and IDRI. Those are the really key organizations that are doing a lot of the screening for the entire consortium. Besides, of course, the two pharma companies that continue to stay in the game as GSK, our colleagues GSK and Sanofi, and you've heard from them earlier today. And then phenotypic screening, which has really been the workhorse of generating lead molecules for TB, can only go so far, and we really need to understand the mechanism of action of these compounds or the targets of these compounds. So the TBD has also invested a lot of resources and know-how and experimentation in understanding mechanism of actions through a variety of tools, resistant generation or genetic tools, et cetera. So really a state-of-the-art collection of capabilities and tools have been brought together and continue to be expanded and developed. And of course, underneath is the entire experience of the pharma industry that continue to still be in the game. I mean, GSK and Sanofi are really involved through preclinical and development, but as you can see, there's a lot of other pharma companies that have joined the organization that bring their expertise from drug discovery perspective, as well as their library, and as well as interrogating the chemotypes that they see, because they've seen so much, and I think that really helps also the organization of the TBDA. So I think this is a great platform that one can build on to go forward. And I just wanted to go over a few of the capabilities, just to give some examples of what we're talking about when we're talking about some of these capabilities. And the first is the screening component. What have we been screening with in the last 50 years? And that's been a question that's been answered recently. And understanding that the bacterium behaves differently, or a drug given to a bacterium drug behaves differently once you give it in different conditions. And here's, for example, various carbon sources. If you look at the bottom box, if you will, you can see that not all compounds work in the same condition. Some work differently, very few work across all the potential carbon sources, for example. And that could be significant when we're looking at an in vivo evaluation for the compound. Do the compounds work in hypoxic conditions, under other variety of conditions? And these are the kind of screening capability and assay expertise that now exist within this consortium. And actually, most places now in the academic environment as well, which is really great. So given that, actually, this is probably an outdated slide already from within the TBDA. As you can see, at this point in time where this slide was made, almost 2 million compounds from the various pharma companies were screened, at least in a replicating condition under glucose conditions. But you can also see they've been also screened in various other conditions, under cholesterol conditions, for example, and slow replicating conditions, which is low pH, for example, et cetera. So this is quite a lot of effort that's gone on into this. And so far, about 200 hits as of this slide were isolated or were identified through the TBDA since 2012. And most of these are being interrogated quite heavily right now as we speak. So that's the screening component of it. I also talked about the biology, which is really, really critical, and some of the tools in biology that really creates a world-class TB biology has also been put together. And I wanted to talk a little bit about some of those tools. And this is a number of, it's a busy slide, but it's a number of steps that we have at our disposal to really dissect out what a compound does for TB. Forget about the top row, because that's really your standard traditional pharma type evaluation of cytotoxicity, physical chemical properties. But in the middle is really a lot of the tools that help us dissect out what a phenotype, what a chemotype does, actually. We have ways of understanding whether a compound is acting on a cell wall biosynthesis, for example. We also have assays that really decipher what the actual target is. For example, we have DPRE1 inhibition assays, et cetera, where we can then classify the various compounds that we have. And you heard about DPRE1 earlier today. We have transcriptional profiling. We have some macromolecular incorporation. I'll give you an example of that. And metabolomics as well, and also a lot of the knockdown mutants that are becoming very invaluable to understanding the target and the mechanism of action of these compounds. So let me give you a couple of examples. And this is an example of macromolecular incorporation. And this is a very simple way of really focusing in on a target, for example. And you've heard about MMPL3 as a target earlier. And compounds that do hit MMPL3 should really start accumulating. If you can see, it's called TMM on the left-hand side. That's trihalose monomycolate. And this is a building block that continues to build up if you inhibit the target. So it's an indication. It's not a complete indication, but it's an indication that the compound may be working on the right target. So it helps the optimization from a medicinal chemistry perspective once you have these kind of tools. On the metabolomics and the chemogenomics component, this is an example of how a knockdown can really be used. In this case, this is an ATP synthase knockdown, a strain that Dirk Schlampinger developed at Cornell. And as you can see, DCCD, which is an ATP synthase inhibitor, dicyclohexylcarbodiamide, becomes much more, so the bug becomes much more sensitive to this compound as you knock down ATP synthase, which means that it's potentially on target. But more interestingly, the bottom bars, the blue and yellow, the beautiful blue and yellow colors, if you will, it's really an indication of using these knockdowns and treating them with alternative compounds, for example, and in this case, nigericine, for example, where you can see the yellow light up. And that means that this strain has become sensitive to this alternative compound, which could potentially mean that there might be some additivity or synergistic activity between this specific mechanism of ATP synthesis and other mechanisms of action. So these are kind of tools that are at our disposal. Other tools that are at our disposal are where the compound potentially accumulates and ends up. With some of the examples that was done by Veronique Dartois at Rutgers, on the left panel shows where pyrazinamide, for example, distributes within the cavity and where clofazimine and bedaquiline distributes. Now, this is not an all-or-nothing type of thing where if you don't see accumulation of a compound in a specific compartment, doesn't mean that it's not a good compound. It means that it's potentially, if you want to have a regimen where you want to maximize your chances, well, maybe you should be looking at the regimens that actually hit everything possible, you know, and not choose things that only hit one thing. So it's a tool that one can use to make some decisions. And the right panel is another tool that's been developed by Curie at Cornell, where he can now look at intracellular concentration or uptake of compounds as far as the outer membrane of the bacteria, which we all know and believe that it's very, very tough to get through. And what he's done through some washing with detergents, he can remove the outer layer of the bacterium, and the bacterium still is completely viable. So it completely replicates normally. So by this differential mechanism, we can see what compounds actually get stuck in the outer membrane and what compounds can get in. Beyond that, actually, which is even more interesting, is we can understand once the compound gets in, does it get transformed to another metabolite? And this is another very, very valuable tool once we start analyzing and doing medicinal chemistry and understanding a compound. And so I want to leave finally with another type of analysis. This is really the breakdown of the first set of compounds. And I think what's interesting here is that almost half of these compounds that are being followed up have still an unknown mechanism of action, which is not your typical mechanism of action like a cell wall biosynthesis or a DPRE1 type of thing. So it's quite encouraging. Once we start understanding what these targets are, I think we're going to start finding really interesting targets. Now, also interestingly, with this set, we now have a large number of compounds that we found phenotypically to be active. I mean, GSK were the first to start this, where we have a large collection of compounds that are phenotypically active. Now we can start combining these with all the genetic validation tools that we have, where we can understand what are the critical targets in TB, and start interrogating from a target-based part, testing these phenotypically active compounds, which we know get into the bug and kill the bug. If they hit this specific target, we already have a match. We can deorphanize a target and start working with the knowledge of the target, potentially X-ray structure, et cetera, which really makes drug discovery much, much easier. Well, not much, much easier, but easier. So, It is hard. So really, this is the basics of what has come up with the TB Drug Accelerator and why the TB Drug Accelerator, the academic groups that are really working on basic research, and the portfolio really maturing in the PDPs really start, my perspective, make us well poised to deliver over the next few years. And I'm not going to dwell on some of the strengths and weaknesses of the TB Drug Accelerator, but which I covered already. But ultimately, it was a bit of an organizational issue that we needed to also address, as how do we best organize ourselves to really deliver, because it is a large consortium of capabilities, individuals, institutions, and pharma companies. And the way that the TBDA is now organized is in teams. Each team is responsible to deliver and chooses to work on specific targets. Of course, there are constant discussions and collaborations and meetings as far as what we are doing. As you can see, the green indicate teams that have pharma companies involved, and the gray are more institutions and PDPs. So, and as I said, Caliber was the last team that joined the TBDA earlier this year. So with that, I want to finish by giving an overview of the portfolio, where it's at. I think you heard about two of these compounds. I know you heard about two of these compounds earlier. The BTZ-043 was discussed earlier. That's in preclinical development. The LUARS compound, the GSK compound, the tRNA synthase compound, that was discussed by David. And then the only other one that's in preclinical development today is the AZA-indole DPRE1 inhibitor that the TB Alliance was collaborating with AstraZeneca, and when AstraZeneca closed their site in Bangalore, we took over the asset, and we are currently doing preclinical development on the lead compound. And this is, again, another DPRE1 inhibitor. However, the difference between this and BTZ-043 or PBTZ, which is now in phase 1, we're going into phase 1, is that these are reversible inhibitors of the enzyme versus the covalent inhibitor of the enzyme, which is exemplified by BTZ-043 and PBTZ. The situation for the portfolio is much better when we go into lead optimization. So the portfolio is far richer. And I don't expect you to read everything unless you have great eyesight. But what I really wanted to show you in a graphical form, how many projects are in that in here, and I apologize if I've missed one or two here. But this is based on some of the information that I had. What I wanted to highlight is in color what sort of programs are in there. And if you can see in the green, these are all novel targets. But these are novel targets in pathways that we know. For example, energy metabolism, cell wall biosynthesis, et cetera. But all of these are novel targets, so there's no pre-existing resistance to these. So that's already exciting as far as the programs that are in the portfolio. On the top two is what I talked about earlier, is to address potential safety, to be a fast follower, if you will, just like the pharma company. We have very good compounds. We now know and understand the effect of a linezolid or an oxazolidinone. What if we could come up with an oxazolidinone that were potentially safer? We heard earlier about the mitochondrial protein synthesis inhibition as being an issue with the class. But what if we came up with a compound that really was almost devoid of that and therefore increased the safety window? So at the TB Alliance, we are working on an oxazolidinone backup as well as within the TBDA. So there are two teams working on it. We certainly are progressing really well, and by the end of this year, we hope to be able to select at least two compounds that we can progress into preclinical development. We have, to the best of our knowledge, dialed out the mitochondrial protein synthesis inhibition, or at least diminished it tremendously. And our 28-day rat study is really indicating that it probably holds true. So that's really showing promise. And the diroquilin is, of course, a follow-on to bedaquiline, and we are also looking at compounds that are potentially could be dosed higher without coming up with adverse effects, especially in cardiovascular events. I also listed in orange, I believe it comes up. Again, addressing resistance. And there are a couple of compounds in the portfolio where we are hitting INHA, but these are these are direct inhibitors, of course, so they are hopefully they completely get around this activation resistance that we see with INH. You heard already about the macrolides and the cyclic peptides and the ureas from Sanofi. Those are also coming very close to at least getting into non-GLP toxicology studies, or at least a decision is going to be made. With GSK, there are also programs where we are approaching decision-making to enter non-GLP studies. The PKS13 also will be advancing very nicely. So bottom line, there are a number of programs that are really advancing well that by the end of this year we'll be in a position to either select to go into non-GLP toxicological studies or make a decision whether we should continue or not. And I wanted to also point out two other things, which I think is also needed quite desperately, and I mentioned at the beginning. In the yellow is a completely novel pathway. The CLIP program is a completely novel pathway. No one's ever attempted to hit proteostasis within TB, so I think that's a really good start to start looking at alternative pathways. And in the gray, I think this is also very exciting. You sort of saw it on David's slide, but I think this is another example of looking at drugs that affect the bacterium in a very different environment, like a different medium. This is very much an interesting concept, and it's very different than anything else we've seen before. And this is also advancing into toxicological studies, so it's quite exciting for GSK. The situation, as you can guess, is better even in the hit to lead. As you can see, the attrition rate is high in the hit to lead, so you need a lot more programs. But this is again color coded, and the color coding I think is it's even more balanced from my perspective. We still have two programs where we have clinically validated targets. And where there are resistance, I mentioned RNA polymerase as a really, really good target that has been clinically validated for treatment shortening. And really, it behooves us not to be continuing to work on such a target if we can get around the resistance of rifamycin. And as you can see now, the yellow is expanded, so there's a lot more novel pathways that are being hit in this category of hit to lead. And we continue to have novel targets. And here, there's another added component. In the dark gray, these are compounds that are actually working in a different state. These are compounds that are working only in a non-replicative state. So it's a completely different, these are not rapidly replicating cells. So within the TBDA and within the investigators who are working in this area, we are trying to sort out how to evaluate these compounds, because this could be also a very important component of the ultimate regimen that we come up with. And of course, there are a number of other programs where we still don't know the target, and we're actively working on finding the targets for these things. And even beyond that, there are a number of programs that are being assessed, as I mentioned here, just 18 projects in formal hit assessment. So when we put it all together, I'm almost done. Here's, without names, the way it looks. I mean, it's glaringly obvious that we're missing something on the right-hand side. And I think this is really the biggest challenge for us. There is an urgency. We need to urgently progress as many compounds from the lead optimization into preclinical development and into the clinic as fast as we can, especially with these new mechanisms of action, these new targets, to really validate these things clinically, because that's still a huge step that we need to achieve. But I hope, I convinced you that the lead optimization portfolio is not too bad, and it's really developing nicely to be able to deliver a number of new programs, a number of new targets that then we can put together in a semi-rational way to see if we can come up with a much better regimen. The early portfolio is rich, as I said, but with those riches, of course, we need to make sure that we prioritize the discovery portfolio very carefully and manage it very carefully to make sure that we put all of our resources on the right ones so that we can deliver faster. If we continue to work on too many programs, of course, as any other organization's funding and resources are not unlimited, so we need to be very clever and careful about how we prioritize and get the work done. So for me, finally, I'd like to acknowledge our the sponsors and supporters as well. And finally, thank you for your attention.
Thank you. Are there any questions?
Thank you very much. It's great to see a progress report from the Drug Accelerator, and that looks very promising indeed. My question really is about the libraries that are being screened, and I was wondering if the TB Drug Accelerator is doing anything to increase the chemical diversity that's being looked at, or is it really just relying on the libraries of the existing pharmaceutical companies?
No, I think they actually are, and there are a lot of other things that that the TB Drug Accelerator is doing in areas of natural products, for example, investing in natural products, and some of the other academic groups that may have alternative chemotypes, if you will. So I think it's not the majority, of course. The majority are the pharma companies' chemotypes, of course, but there is an effort ongoing, yes, to start looking at alternative chemotypes.
Any other questions or comments?
Marina Protopopova, Sequala. I'm just wondering how intellectual property is handled through your process, because it's quite a substantial amount of data generated. So who has?
So that's a very good question. Actually, it's interesting because it's starting to get to a point where we have to worry about intellectual property, very, very, very much so, especially as we go into lead optimization. Before that, I don't think it's really that important because the attrition rate is so high. And we are starting to really experiment on a one-on-one basis between collaborators. We have experience with GSK, we have experience with Sanofi, GSK and others are starting to have experience with each other. So I think this is starting to evolve. And I think it's going to be case-by-case basis.
Any questions on this side?
Thanks. I also appreciate the overview. One question I have, when you show the slide that you've screened about 2 billion compounds, do you have a library where this is accessible to avoid duplication of effort? Others should know what's been tested under what conditions and not embark into doing something that's already been proven wrong.
So of course, the pharma company compounds are proprietary, right? So they are blinded when they're screened. And the hits that come out are still blinded until a pharma company decides that they want to disclose that to the entire organization. However, in the background, there have now been developed algorithms where you can actually look at a set of unblinded compounds to really, from fingerprinting, to see if similar compounds have been tested, or very, very similar compounds or identical compounds have been tested, and therefore not duplicate things. So that can be done in the background.